Hi friends, welcome back to my channel Only Bio. Continuing with the series of Do You Know? Now, today we shall understand something about genetic modification. The question is, what is the genetic modification effected by the DNA of the virus termed as? So we need to understand the process, and the process is called as transduction. Okay, transduction is the process by which foreign DNA is introduced into a cell by a virus or viral vector. A virus and viral vector, why is it being written like this? Because if it happens with the virus, that can be natural. But when we are using the virus artificially, we call it as a viral vector. Here, vector means an agent. So an example is the viral transfer of the DNA from one bacterium to another, and hence an example of horizontal gene transfer. Transduction does not require physical contact between the cell donating the DNA and the cell receiving the DNA, which occurs in conjugation, and it is DNase resistant. That is, transformation is susceptible to DNase enzyme. You might be aware of that DNase is the enzyme that can denature or act on the DNA. Transduction is a common tool used by molecular biologists to stably introduce a foreign gene into a host cell's genome, that is both bacterial and mammalian cells. In this picture, you will get an idea about the transduction process, which has been numbered from 1 to 7. Now, in the first one, that is number 1, you can see that the bacteriophage, that is the virus that infects the bacteria, is trying to enter the bacterial cell, which has its own chromosome, right? In the second step, we find that the phage has inserted its DNA, and at the same time, meantime, we find that the bacterial DNA is literally being sliced. In the third pic, you can see that the virus has taken up the host DNA and it starts producing its own copies. And in so many small viroids, or rather the small new viruses that have been created, you can see one new one, okay, which is a defective fudge or a defective one. It's actually a change one. In the fourth one, we can see that this defective one has been selected up, and this one, okay, the defective one is the one which is now infecting up a new cell, and it is introduced its DNA, which also has a part of the donor DNA. And this is how, once it inserts it, because this has been act as a vector, in the seventh series, or in the seventh pic, you can see that the bacteria has undergone a change. It is not only having its own DNA, but the darker part indicates that this is a foreign DNA. अब ये foreign DNA आया कहाँ से? तो this has been picked up by the host DNA, that is the donor DNA. Friends, hope you have understood this and hang on with me for many more. Thank you so much.